Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Roslin, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. Please enter your questions at any time. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Jeroen from Rousselot.
Hello, everyone. Thank you for joining this webinar on vaccines. My name is Jeroen Geeraerts, and I will be hosting this webinar today. I work as a business development manager with Rousselot, specifically for our biomedical segment. The topic of today is optimizing your excipient screening for vaccine formulation with an ultra-pure pharmaceutical gelatin. This presentation will be divided into 3 parts. We will spend most time talking about vaccine types, formulation of vaccines, and the benefits of gelatin can bring to this. Next, we will go into more depth of the process of making pharmaceutical-grade gelatins and explain how we make the difference with our unique range of low endotoxin gelatins branded under the Xpure name. Lastly, we want to touch upon Rousselot's global presence. With more than 125 years of experience, Rousselot is the leader in collagen solutions. We are part of a bigger company, Darling Ingredients, which is a stock-listed US company. Needless to say, should you have any questions after attending this webinar or would like to receive more information about our Xpure offer, you can always find more information via our Biomedical website or contact me via mail directly. On a practical note, we will not be answering any questions during or after this webinar. However, as mentioned, feel free to contact us and we will be more than happy to answer your question or accommodate your request. As the world is currently undergoing the COVID-19 pandemic, there have been many to draw parallels between this pandemic and the one the world faced more than a century ago. Looking back, 2018 marked the 100th anniversary of the deadliest pandemic known to date, more commonly referred to as the Spanish flu. It was estimated that 500 million people were infected, which constituted 1/3 of the world's population at the time, with a death toll ranging anywhere from 17 million and possibly even as high as 100 million people. Today, less than 2 years later, we are confronted with another pandemic. I'm sure that many of you have seen Bill Gates' 2015 TED Talk about facing the next global viral outbreak and how, as he said, the world was not ready for it. Unfortunately, in retrospect, he was right. Of course, the most powerful tool we have at our disposal to fight this pandemic is, of course, a global vaccination program. A relevant example of this in modern history is smallpox. This virus used to kill millions of people every year and was declared eradicated in 1980 following a global immunization campaign led by the WHO. Going over to this slide, I'm going to talk about the different vaccine types, of which there are 4. The first category are the live attenuated vaccines. They are made from a weakened form of the pathogen. 2 known examples here are measles, mumps, rubella vaccine and the varicella or chickenpox vaccine. The second category are the inactivated vaccines. They are made from the dead form of the pathogen, and 2 known examples here are hepatitis A and rabies. The third category constitutes the subunit or recombinant form. These can be made from a part of the spike protein of COVID-19, for example, or the RNA or DNA of these pathogens. Hepatitis B and the human papillomavirus are 2 examples. As the last category, these are designed on the toxic compound that is released by the pathogen known to cause the disease, which is, for example, in the case of tetanus or the diphtheria vaccine. These are called toxoid vaccines. Now, the main differentiator between category 1 and 2 and category 3 and 4 is the need for stabilizers in these formulations. These are typically added to maintain the viability or stability of the virus or pathogen during the manufacturing process. As an example, gelatin is one of these examples that has a long-standing track record as a vaccine stabilizer. In this webinar, we have already touched upon the role of the World Health Organization briefly regarding the smallpox vaccination program during the 1980s. Of course, it acts as the body of the United Nations responsible for coordinating health globally, and again today plays a crucial role in the COVID-19 pandemic. The WHO has drafted the landscape of COVID-19 candidate vaccines, which is updated regularly. Day 15, they reported 110 candidate vaccines in preclinical evaluation and 8 candidate vaccines in clinical evaluation. In an effort to return the world to where it was before COVID-19, we can only hope many of these candidate vaccines can prove their safety and efficacy via clinical studies. Now, linking back to the 4 different vaccine types from the previous slide, it's worth mentioning that 11 of these candidate vaccines are using the inactivated or live attenuated vaccine approach. Zooming in on these 2 types, we've already learned that the development approach is different. For example, the inactivated vaccines use a dead form of the pathogens, whereas the live attenuated vaccines use a weakened form of the pathogen. So for the first category, pathogens are inactivated and killed by heat or chemicals to reduce the virulence in order to prevent infection from the vaccine. They are classified depending on the method of this inactivation. In this category, a disadvantage is that multiple booster injections may be required to provide an effective immune response against the inactivated pathogen. Thus, attenuated vaccines, so the second category here, are often more preferable for generally healthy people because a single dose is often safe and very effective. However, some people cannot take these attenuated vaccines because the pathogen poses too much risk for them. And for example, elderly people or people who are immunodeficient are a risk group here. So that is also relevant for the COVID-19 virus. The second category, the live attenuated vaccines, contain live virus particles, but they have very low virulence. So when administered, they give a quick response. There is a continuous presentation of antigen, and this creates a durable immunity. So booster shots are required less often. And of course, a benefit here is that all phases of the immune system are activated. For instance, immunoglobulin A, so local antibodies, are produced. A disadvantage, but this is only in rare cases, natural mutations can occur, and this can reverse the virulence. These vaccines, as mentioned before, cannot be administered in immunocompromised patients. Another disadvantage here is that these live strains typically require high maintenance, such as refrigeration and fresh media, making transport to remote areas difficult and costly. Talking about inactivated and live attenuated vaccines, we now know that these 2 types of vaccines require strong stabilizers. Now, there are 5 categories of stabilizers available, ranging from amino acids, antioxidants, proteins, sugars, and surfactants. Now, of course, the final formulation will always be a combination of these, which is best suited to that specific vaccine. But then the question, of course, is how is that determined? First, let me go into key requirements for vaccine stabilizers as general. So they have to be, of course, biologically safe for in vivo use. They have to have stabilizing properties. They have to be readily available and easy to use, and they have to have minimal side effects. Now, going back to the composition of these stabilizers, historically, this excipient selection was mostly done empirically. So, of course, successful vaccines were developed in the past, but they were always very reliant on cold storage and limited transport. Now, today, newer formulations are luckily more scientifically driven. So, because there is a better understanding of the virus structure, but also the immune system regulation, this enables a more informed decision on which stabilizers to use. It's important to note that the type of vaccine is still the main determinant of which stabilizer is being considered. And as a general note, most stabilizers are selected from a range of generally regarded as safe excipients, of which gelatin as a protein being a very good example. So gelatin is a biological macromolecule that serves as a versatile drug, or in this case, vaccine delivery carrier, and is also categorized as a grass excipient. As mentioned, gelatin has been a trusted excipient in the pharmaceutical industry for decades. So on the right-hand side, we have listed commercially available vaccines that use gelatin as an excipient. It's worth mentioning that not only vaccines that use the parenteral administration route use gelatin, but also vaccines that are administered nasally use gelatin. So the first vaccine listed here is the Fluence Tetra from AstraZeneca, and that is a vaccine that uses gelatin as well. But of course, what is now the crucial role that gelatin as a stabilizer can play in these vaccine formulations? So We know for the inactivated vaccines and the live attenuated vaccines that they contain whole pathogens. So these are larger molecules that are not stable on their own and require larger proteins to stabilize the formulation. Gelatin, being one of these, has these unique properties to stabilize the larger pathogens. Additionally, it's worth mentioning as well that gelatin is difficult to replace in the freeze-drying process. Going to the third category, the nasal vaccines, here gelatin works as an adsorbing stabilizer to retain the immunization properties of the vaccine. As mentioned with the fluenza tetra example, gelatin has already proven able to fulfill this function as it is commercially launched already. In conclusion, we can say that gelatin helps to preserve potency and effectiveness of vaccines in transport, storage, and administration. We have talked about the benefits of using gelatin in your vaccine formulations and would like to introduce our Xpure range, the first complete range of low endotoxin gelatins and collagens. Our Xpure offer is built on 3 pillars: the highest purity, quality, and consistency of the material. In the Xpure process, the purification steps, drying, grinding, and packaging of our low endotoxin gelatins is executed in a GMP-approved facility under clean room conditions. I would also like to point out that the endotoxin levels below 10 endotoxin units per gram, which is the limit of detection for these impurities, is very relevant as all parenteral administrations should be designed to limit bacterial endotoxins according to the USP. As shown in the previous slide, the production of our Xpure range is a unique process patented by Rousselot. In comparison to other pharmaceutical-grade gelatins available, the Xpure process has an additional purification step built in for ultra-low endotoxin and pyrogen activity. As a result of this unique process, we can produce the purest hydrolyzed gelatin available. So that is a type 8 process to be complete. Finalizing this webinar, we wanted to touch upon Rousselot's global presence as the leader in collagen solutions. As a part of Darling Ingredient, Rousselot has more than 125 years of experience with gelatin and operates globally via 11 plants and 9 sales offices, as you can see here on the world map. With our biomedical segment, we focus on parenteral formulations such as vaccines, hemostats, and also looking to the future with regenerative medicine applications. Should you have any questions or would like to receive more information, you can always visit our Biomedical website or contact me via mail directly. Thank you for your attention, and I hope you enjoyed this webinar on vaccines. Thanks,
Jeroen. And thanks to our audience for joining us. You can submit your questions to Jeroen at the email address listed here. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.